Let's discuss question number 3, part A. Figure 3.1 is a photomicrograph showing part of a transverse section through the root of an iris, Iris germanica. Irises are herbaceous monocotyledons. These plants have the same transport tissues as the herbaceous dicotyledons, but the transport tissues are distributed differently. In monocotyledons, the central tissue in the root is parenchyma or packing tissue. So guys, over here we can see that this is the transverse section of the monocot root. This is not a dicot root, this is a monocot root, or we can say root of herbaceous monocotyledon, that is iris, right? So they're saying that the monocot root has the same transport tissues as the dicot root, but they're arranged in a different pattern. So these are the central tissues of the root, right? This is the cortex over here, right? These cells are cortical cells, and we can see central tissue over here. And you know that this central tissue is different from the central tissues in the root that you have studied. You have studied dicot root, right? So only arrangement is different. Tissues are not different. The arrangement of cells are different. So what is R over here? R is basically endodermal cell. R is endodermal cell, right? R is the endodermal cell, and this entire layer is the endodermis, because we all know that in the dicot root, the boundary of the central tissues is the endodermis. So as they mentioned that in the monocot root, the transport tissues are same, right? But only the arrangement is different. So over here we have the endodermal cell. R is the endodermal cell. S over here, the large cell, is the xylem cell, or we can say xylem vessel element. And the T, the small cell over here, is the phloem cell, or we can say sieve tube element. Alright. Cells R, S and T in figure 3.1 are found in different tissues. Name the tissues in which the cells labeled R, S and T are found. So tissue in which the cell R is found is the endodermis. The tissue in which cell S is found is the xylem tissue, right? Because S is xylem vessel element. And the tissue in which the cell T is found, T is sieve tube element. Sieve tube element is found in phloem tissue. Part 2. Outline the role of tissue in which cell R is found in figure 3.1. So basically they want to know the role of the tissue in which cell R is found. Cell R is found in the endodermis, right? And they're asking us to state the role of endodermis. So you all know that endodermis has passive cells which have Casparian strip, and that Casparian strip blocks the apoplastic pathway. So that apoplastic pathway is converted to symplastic pathway. The cell wall to cell wall pathway is converted to cell pathway, right? This is the purpose of endodermis. So what is the role of endodermis? What is the role of endodermis? We can say that endodermis contains passive cells which have Casparian strips. Casparian strips block blocks apoplastic pathway and keep the symplastic pathway open. So ions flowing, or you can say water and ions flowing through cell walls now enter the cytoplasm of the cell, right? So basically the endodermis blocks the apoplast pathway and converts the apoplastic pathway into symplastic pathway. This controls the amount of ions or the type of ions flowing into the xylem vessel. You can also mention that this controls the flow of ions into the xylem. All right. Let's move on to part 3. State an example of an organic compound that is translocated in the roots of an iris. So they're talking about translocation, right? So which organic compounds are translocated? Translocation occurs in phloem. So organic compounds that are translocated are sucrose or amino acids. So you have to state one example of that organic compound. So we can over here write sucrose. We can also write amino acids. They're just talking about organic compounds. We cannot write mineral ions or we cannot write water. They're talking about organic compound, right? Okay, part B. The electron micrograph in figure 3.2 shows a section through some root cells in an onion allium cepa. So these are the cells taken from the onion root, right? And these are basically onion root cells. On figure 3.2, draw a label line and label it with the letter P to identify one plasmodesma. So we have to identify and label one plasmodesma. So guys, this is plasmodesma. Plasmodesma is basically a strand of cytoplasm that runs through the holes in the cell wall. And basically plasmodesma connects the adjacent cells. So this is plasmodesma. We only have to identify and label one plasmodesma. Plasmodesma is singular, plasmodesmata is plural. Part C. Table 3.1 contains information about 4 polysaccharides found in animals or plants. Complete table 3.1 by filling the missing information. Alright, so we have to state the monomer of amylose. We all know that amylose is made up of alpha glucose. So the monomer of amylose is alpha glucose. What bonds are found? What type of glycosidic bonds are found in cellulose? In cellulose, there are beta-1,4 glycosidic bonds, right? So in cellulose, there are beta-1,4 glycosidic bonds. What is the function of the glycogen? So the function of glycogen is energy storage, but in animals. So over here, we will write energy storage in energy storage in animals. Glycogen stores energy in animals, whereas amylose is store energy in plants. So guys, we are done with question number 3. Now we will move on to question number 4.